Yes, good morning, good afternoon, good evening, good whatever time it is that you are watching this video. Today I'm going to go over some cell organelles. Riveting stuff, I tell you. Uh, Organelles are really cool in that they're really tiny, but they do really specialized things. Right? The development of organelles was critical to the advancement in the function of cells. And without organelles, we would not be able to be the multicellular, awesome, cool, very attractive, in my case, beings that we are. Right? So I just want to review what they are, a little bit of their structure, and a little bit of their function. So let us start with something that all cells have, which is cytosol. So cytosol is the jelly stuff that fills up most of the empty space within the cell. Okay? It's primarily aqueous, meaning it's made out of water, okay? But it is highly concentrated with a bunch of the macromolecules that we've talked about. So like there's a bunch of carbohydrates floating around, a bunch of nucleic acids floating around, a bunch of lipids floating around. And there's also the ions that are necessary for living functions like sodium and potassium and chloride. Okay? So all that stuff is just floating around the cell, waiting to be grabbed up by organelles or other subcellular components for use. Okay? Then we have microtubules, okay, which are protein-structured filaments. And so basically they're little tubes, hence the name microtubules. And you can have one little tube connect to a bunch of other little tubes, and they build up into these tube structures. Like if you imagine you put a bunch of straws around each other, right? That's basically what a microtubule looks like. And then if inside of those straws you had littler straws, and you could telescope them out. The function of the microtubules is primarily for structure within the cell. It maintains the shape of the cell, keeps things where they're supposed to be at. Yeah. Then we move on to ribosomes. A very sexy little organelle. The ribosomes are so cute, so fantastic. The ribosomes are responsible for building proteins, right? It's where proteins are actually made using the information stored within our RNA, messenger RNA specifically. And so the structure of the ribosome is actually comprised of two components. There's a large subunit and a small subunit. And they're made out of proteins and RNA, and the big balls. And they're kind of like hamburger buns. And the large subunit's the top hamburger bun, and the small subunit's the smaller, or rather the bottom hamburger bun. Right? And when they combine, which happens in the presence of an mRNA, they become an adorable little ribosome ready to make proteins. We'll talk about them at more length when we get to protein synthesis, where we actually talk about making proteins. But in general, the large subunit has 3 chambers in the top where the mechanisms occur, and the bottom subunit has the mechanisms for moving along the mRNA. Right? So then we have the last of the every cell has these things, right? And that's the cellular membrane. Of course, you can't have a cell without an external membrane, right? Otherwise it's just the environment. The cell's defining feature is that it's isolated from the environment and has its own little environmental conditions. Right? And so this cellular membrane, sometimes called a plasma membrane, is what divides the inside of the cell from the outside of the cell. It's made from a phospholipid bilayer. 2 layers, bi prefix means 2. And the exterior side of the layers are hydrophilic, meaning they are cool with water, they don't mind water, whereas the interior layer of this membrane is hydrophobic, meaning they hate water. Get out of here, water. They hate all polar substances. This is going to come into play when we talk about cellular transport mechanisms. There are also proteins embedded within these membranes, either all the way across, which serve as channels, or just on one side, which serve as communication mechanisms, or places where enzymatic reactions can occur. Important to note that the inside of the cellular membrane can actually be folded up, okay, increasing the surface area of that region. So it allows for different chemical reactions to occur in lieu of having organelles. So all cells have those things that I just talked about. Moving forward now, we are going to be talking about specifically membrane-bound organelles. That is to say, organelles that have their own membrane around them. These are unique to eukaryotic organisms. Okay? The defining feature of a eukaryotic organism is that they have membrane-bound organelles. If you don't have membrane-bound organelles, you're a prokaryotic organism. Cool. So the first of these is, of course, the nucleus. It's a prime example of a membrane-bound organelle. Effectively, the nucleus is just a ball of membrane chilling out in the cell that keeps the DNA safe. The membrane that surrounds the nucleus is called a nuclear envelope, and it has pores in it, holes, so that the RNA can come and go and the necessary nitrogenous bases can come in and come out. Right? Nucleus. Like, it's not a fancy thing. It's just basically a house for DNA. The nucleolus is a region within the nucleus where ribosomes are constructed for eukaryotic organisms. Obviously, prokaryotic organisms do not have nucleuses, and so their ribosomes are generated out in the cytosol. But it's just more efficient to have a specialized area to create the ribosomes in, and so it's cuter the way that the eukaryotic organisms do it within this nucleolus. Ooh, we got big letters. Go back down to small letters, please. Thank you. Then, adjacent to the nucleus, we have the endoplasmic reticulum. And the endoplasmic reticulum is a big, foldy wall of membranes. Right? It kind of abuts up against the nucleus like this, and foldy, 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 foldy. Anytime you see big foldies, it's to increase surface area. The increased surface area allows for more chemical reactions or more workspace to do things. The endoplasmic reticulum, hereafter referred to as ER, is divided into two subsections. The rephia, which is studded with ribosomes. That region is for making proteins. Okay. The smooth ER does not have those ribosomes. It's just the wall of lipids, and that is used to make lipid products. Okay? So like the phospholipids are generated here, stuff like that. Lipidy things are made in the smooth section of the ER. Talking about smooth things, we got the Golgi apparatus. So this does not live far from the ER. This lives pretty close to the ER. It's this big, foldy, cute vase-looking thing right here. And two sides, you have the cis side and trans side. And this big globby guy is responsible for the packaging of proteins and other things that need to be secreted by the cell. So the protein comes out of the ER pretty well structured. Right? Sometimes they need a little bit more finalization, and that can occur within the Golgi apparatus. They have regions of embedded proteins within the Golgi apparatus, which will interact with new proteins. And if they need to be refolded to meet their final stages, it can do that there. But the primary function is to take proteins or other molecules that need to be secreted from the cell, that is to say, they need to leave the cell, and wrap those bad boys up in a specialized vesicle with little signaling molecules, signaling pieces on them that tell the other cells in the body where this vesicle is destined to go. And so if I'm making a protein in my liver that needs to go to my stomach cells, the Golgi apparatus will wrap that protein up in a ball of membrane. It will stick on some carbohydrate signals that are only going to be accepted by stomach cells. They hit a stomach cell, the stomach cell says, cool, this is for me. It grabs that vesicle, opens it up, takes that protein, says, yay. Cool. Golgi apparatus. Which segues neatly into vesicles, right? I dropped that word a couple times describing the function of the Golgi. Vesicles are just little bubbles of membranes. That's it, right? You take something, you wrap it up in a membrane, that's a vesicle. It's good for transporting stuff around the cell or between cells. Vesicles can combine with other vesicles or combine with other membrane-bound things. So that's how, like, the stomach cell in my prior example would take in this thing. The vesicle would combine with the membrane of the stomach cell, blah, suck it in. Okay. Very similar to vesicles, or rather, I'm stuck here. Okay. Very similar to vesicles is vacuoles, which I just called vesicles again. This should say vacuoles, Vacuoles, vacuoles. They're like big vesicles. The most important defining feature between vesicles and vacuoles is not the size, but the fact that vacuoles will not combine with other membrane-bound stuff. So like if two vacuoles bump into each other, they won't combine to make one big vacuole. They stay very independent from other things. So the vesicles are good for transport if it's temporary, whereas the vacuoles are good for long-term storage. Long-term storage. Cool. Again, this should say vacuoles, not vesicles. Moving on, we have the mitochondria, which is a double-layered membrane. The fact that it has two layers, an outer layer and an inner layer, is very important to its function, which we will explore in more depth when we talk about cellular respiration, which is not far off. We'll have a special lesson on just cellular respiration. So we'll talk about this in detail then. But the mitochondria is the location of aerobic oxidative phosphorylation in eukaryotic organisms for cellular respiration. Okay? Then we have the plant-specialized chloroplast, not just in plants, in some algae as well. We got the chloroplast, which is what eukaryotic organisms use to do photosynthetic reactions, right? So all of the photosynthetic reactions occur within the chloroplast for eukaryotes. Prokaryotes don't have membrane-bound organelles, so they don't have chloroplast. Okay. So the chloroplast is similar to the mitochondria in that there's an outer membrane and an inner membrane. One of the huge differences, though, is that the chloroplast's inner membrane has been specialized into these cute little stacky structures called thylakoids. And that's something that we'll talk about in depth when we look at photosynthesis. Again, not far off. Okay? Moving onwards, we are almost done here. Okay? We got centrosomes. So centrosomes are specialized microtubule structures in animal cells. They look kind of like celery in the models of them, but their job is to move chromosomes during the process of mitosis or meiosis. So they make sure the chromosomes go to the right place at the right time. We will talk about that at length when we look at cell division. Plants don't have centrosomes, so instead they utilize a specialized type of vesicle to move the chromosomes. Okay? Is that it? Is that it? That's it. That's it. That's it. That's everything. So cool. All right. Bye.